Institute for Bioengineering of Catalonia in Barcelona, Spain. I'm going to present our research done in implantable sensors for ischemia detection. So first, a little bit of introduction of our nanobioengineering group. This is a highly multidisciplinary team. We are chemists, physics, biologists, engineering, with three main different areas in the group. So let me put the laser. So there is the area of nano micro environments that is mainly focused in 3D co-culture of cells for mimicking different biological models. We also have the area of microfluidics that is also integrated with the other areas, and the area of biosensors and point-of-care devices, and also the conjunction of these three different areas. We are doing point-of-care devices, lab-on-a-chip systems, and organ-on-a-chip systems for mimicking advanced models. But I'm going to focus more in the biosensor area. Okay, so first I'm going to introduce the problem that these sensors, these implantable sensors, are focused in the detection of ischemia. And ischemia is basically a lack of nutrients and oxygen into the tissue. So here you can see a scheme of a cell. If we are reducing the presence of oxygen.
Sorry to interrupt. The slide is not moving.
Okay. So it's a problem because I don't know if I stop to share the presentation. Maybe, no, but then I cannot. Well, then let me.
Yeah, can you stop sharing
Show it
and
in
share?
this way.
Because
Okay.
I only see your first page, bro.
Okay, then I show in this way and then I follow with the presentation. Okay, so basically in the cell, when we are reducing the oxygen, we are stopping certain activities into the cell, like the generation of energy, ATP. If we don't have so much energy into the cell, the pumping of the ions through the cell membrane are not working. So we are also changing the concentration of sodium and potassium. And also, if we don't have oxygen, we are not having an aerobic respiration. So we have an anaerobic, and then it's producing.
Monica, we are unable to see your slides. I mean, we are only seeing your first slide. Can you move your slide? Next one.
Well, in fact, I'm moving the slides.
Yeah, but we are only seeing your first slide.
So I don't know which is there is any way to change the.
Yeah, can you just stop sharing and again share it once again?
Okay. Or maybe it's a question of the configuration of the presentation.
Yeah. Can you share it again now?
Okay. Sorry, espera. Wait a second. Okay, can you see the.
It's loading.
Can you see the slide?
No, we are seeing the conference page. I think you have to share your screen, entire screen. When you are sharing your screen, you have to select the option entire screen.
Yeah, but it's not appearing the presentation when I pressing the sharing screen. It's not appearing the one from the presentation in the PowerPoint. So it's appearing all the things except the one from the presentation. So wait a second. Maybe I can do it in this way.
Yep.
Now it's fine.
It's loading. Yeah, I can see. We can see your screen now.
Okay, thank you. I'm sorry. Sometimes,
no problem.
Okay, so basically it's the presentation of the different areas of the bioengineering group, and here I can show you a representation of the problem of ischemia. As I commented to you, this is the representation of a cell, and when we have a lack of oxygen, then it's okay. When we have a lack of oxygen that we can detect it with an oxygen sensor, ATP is affected, then the pumping of ions is also affecting. Then we can detect this change of ions pumping with a potassium sensor, for example, or sodium sensor. and also is changing the aerobic respiration of the cell. Then we are producing more acidic solutions into the cell or in the surroundings of the cell, and we are detecting with a pH sensor. Okay, so basically the problem of ischemia in the medical diagnosis is something that could be detected in around 5 to 8 days after the ischemia happens. So that's, and theoretically the degradation of the tissue is happening in just 2 days. So when we are detecting the problem, the tissue is already degraded and cannot be recovered. So the point is to develop a system that permit a fast and easy monitoring of ischemia in a fast way. So the current methods that are available in the market is, for example, the Cook-Schwarz Doppler, in which there is a sensor surrounding the arteria. So it's quite risky, the implementation of these sensors, and also has high false positives. Also, we have the strict needle, that is a kind of needle that you can insert it into the tissue and measure the change of pressure. So it's an indirect detection of the lack of oxygen, and also has not a continuous monitoring. And also the microdialysis, that is detecting the ions, this pumping change of the ions, also by extraction sample, so it's not also continuous and has also certain errors. So with the hospital, the key opinion leaders in different hospitals that we collaborate with this project, they commented that they test all this technology, and the final gold standard that they are using is the frequent monitoring of the tissue. And when they realize that the tissue is started to be a bit warm up, they know that is something happening into the tissue, but that's detected after certain days that happen the problem. And these kind of ischemia problems are affected if you have an hemorrhage. So, for example, laparoscopic or robotic surgery is necessary to detect this problem. Also, in the surgery of flaps, when you are transplanting a tissue in different areas of the body, or in the compartmental syndrome that happens when there is a trauma and block the flow of the blood in this area of the tissue, or with fetal growth restriction when the fetus is not pumping with the required nutrients. So some numbers behind part of these medical problems, for example, reparative surgery, where the flap surgery is required. Just in United States, there is around 200,000 patients per year. And the second surgery that require the problems in ischemia, it doubled the cost of its surgery. Also in compartmental syndromes is affected around 3 million patients in United States, and it has an extra cost of 2,000 million euros per year. And that's just two of the medical diseases, that there are many others I commented before, and that's just in United States. So in the whole planet, these numbers are much higher. So the technology that we propose is a technology that could be implanted directly with a needle inside the tissue. So it's a micrometric sensor that could integrate more than one sensor, and is an array that initially we developed for different sensors that could be just implemented one type of sensor. And the idea is to send this signal in a wireless method to a mobile or a computer. So basically, the technology that we propose permit a continuous monitoring of ischemia inside the tissue, and also it permit the analysis inside the blood. It's a miniaturized system that could be inserted with a needle at any part of the body. It's flexible and has an easy manipulation and easy interface, has no risk in the implementation, and has a low cost. So comparing with the technologies that I already introduced that is commercially available, if we compare with the flat microdialysis, let me put the pointer. With the flat microdialysis, we have a faster detection and is real and continuous monitoring. Also with the Doppler, we don't have the risk that has the Doppler, and comparing the cost, our technology is much cheaper than the current methods in the market. So we are going to propose to develop three different sensors for detection of ischemia in tissue, so based on oxygen sensor and potassium and pH with a ion selective sensor. So here, the picture of our technology, this is the type of sensors that we are working with, and this is the electronics that we are developing. That's a microelectronics at the moment. It's not the miniaturized one, but basically for the development of our sensors for the in vivo analysis. So the introduction of our pH sensor is an ion selective sensor based in this polymer, that is the polypyrrole. This polymer, as you can appreciate, is affected by ions, about protons, so we can deprotonate and protonate this polymer, and it can be electropolymerized. So just applying a voltage, we are able to polymerize this polymer on an electrode. That here is a same image of the electrode before electropolymerization and after polypyrrole immobilization. So we have detected, we make a calibration curve with standard concentrations of protons of different values of pHs in the ranges of the in vivo modification of ischemia. And we also detected with different ions as potassium and sodium in order to see the selectivity of our sensors. And also we detect the evolution of these sensors for months. And as you can appreciate, the sensitivity of the sensors is not changing under in vitro conditions. This is the performance of our sensors. So the electrochemistry that we are detecting is this change. So the oxidation, so the reduction of oxygen to water, and the electrons that we are producing in this reduction, we are analyzing in our potential state. So here you can appreciate the values of oxygen on the different conditions of oxygen. So from low concentrations to higher concentrations of oxygen, we have a higher reduction with the bare platinum and with the platinum modified with Nafion. Here you can see different response of this sensor and the different protections layers of polymers. And also you can see here in front of time, which is the evolution of our sensor, if we are reducing the presence of oxygen or increasing the presence of oxygen. As you can appreciate, is a reversible sensor, so we are able to analyze in continuous different concentrations of this oxygen. We also tested our sensors and the sterilization with UV, and the signal is not changing so much. It's changing the starting value of background, but we can normalize this value. And the difference when we are applying the changes on pH is not, this jam is not changing so much after sterilization. That is quite important in implantable sensors. And also you can see the good stability of the sensor reusability. So how many times we can reuse the sensor in front of time. It's around 1 month that we are able to reuse the sensor. Then after the optimization of this sensor under in vitro conditions, we analyze it in vivo. So the two different models that we develop is with in a rabbit model to analyze the muscular tissue of these sensors, the ischemia in the muscular tissue. And also in order to analyze fetal growth restriction, we analyze a model of fetus in a sheep model, much bigger, in order to have a similar model to humans in a sheep model. So we start with a rabbit model, and first we anesthetize the animals, introduce with a needle the sensor in the tissue, in the muscular tissue, and then we control the breathing of the animal with this equipment. So basically we can know how much oxygen.
Monica, again, your slides are not moving.
Yeah, okay. I don't know why it's happening. So you cannot see my screen anymore?
We can see your screen. We can see your third slide of the presentation.
Okay.
Can you put it in the presentation mode?
It is in the presentation mode.
Yeah, now we can see your slide number 15. Can you go
Okay,
up
so
and down?
yeah, but if I put
Yep.
in the presentation mode, you cannot see the slides then.
hmm Okay,
Okay,
fine.
then
Now,
I left it in this mode and would be
can you
easier.
increase the zoom? Can you increase the zoom?
Yeah, a little bit and left it in this mode. Yeah.
Yep, fine.
Okay, so this is the in vivo steps with the rabbit model. So basically, we control the breathing of the animal with this equipment in order to know which is the amount of oxygen that is breathing the animal. Then parallelly, we are extracting blood and analyzing also with a standard equipment the blood of the animal under normoxia and hypoxia, and we continuously monitoring the tissue, the concentration of oxygen and pH. Here you can see the results with the blood analysis of this extracted blood from the animal under normoxia and hypoxia. We can see standard statistical differences between the two stages. And however, when we are measuring pH, we are not able to analyze the recovery. So when we are going back to normoxia into the animal, we are not able to see the normoxia state, basically because the animal is producing lactic acid and is not removed so fast from the tissue and from the blood. But basically, this is the linear response of this hypoxia and normoxia states. We are also able to detect the same in the in vivo animal. So in a state of blood extracted from the animal, we are able to see also differences between normoxia and hypoxia. And in this case, there is also no difference with the recovery. So the linear response is similar to the ones that we had measured in the blood of the animal. And then we move from pH to oxygen. And with the oxygen, the response of the animal of the sensor is quite different. As you can appreciate, we can see high differences from the normoxia to hypoxia. In this case, we are detecting directly the oxygen that we are injecting inside the animal. So it's not a indirect detection, and it's not producing anything that need to be recovered. That's why the recovery stage of the animal can be so well detected in the oxygen. So it seems that in oxygen is working better than in pH for these reasons, because we need to remove the lactic acid produced. This assay is analyzed in blood. And in tissue, we have similar responses, but the recovery is not so clear in tissue. Maybe because the diffusion of the oxygen in blood is much faster than in tissue. Then in blood, we are able to detect it quite well. But at least the difference between normoxia and hypoxia are quite high, much better than in pH. So we move to a different model. In a sheep model, in order to analyze fetus growth restriction in fetus, so what we have done is to insert our sensors inside the muscular tissue of the fetus on a model of a sheep. And what we are doing was to insert our sensors inside the fetus, and then we control the hypoxia by blocking the umbilical cord flow. So what is happening in the normal models that has growth restriction, that the problem is in the umbilical cord, and it's not possible to flow so well the nutrients to the fetus. Then also we extract blood from the fetus, and we are monitoring continuously into the tissue. So here you can see the values of pH in intramuscular, so inside the tissue, and intravascular, so that we are able to inject one of our sensors inside the vessel of the fetus. In fact, working with fetus model is much more challenging because obviously the size is much smaller, and also the changes on pH and oxygen are much smaller than in the adult. So the differences are more small and more difficult you need a more sensitive sensor for detecting. In pH, as in the previous model, we are able to see the difference between normoxia and hypoxia. And in this case, what we was measuring is 50% of the occlusion of the umbilical cord or 100% of the occlusion. No big difference between this occlusion, but clear difference with normoxia. So we are able to analyze this hypoxia. And in the vascular, in the blood, we are more able to analyze this recovery. In the pH, better than in the previous model. In fact, that was not so clear. The recovery in fetus in blood is easy to detect this recovery. And in muscular tissue, the detection of oxygen for this fetus was maybe not so clear as with the rabbit, but we were able to analyze certain differences, mainly with the 100% of occlusion of the umbilical cord. And yeah, we was able to detect the difference between normoxia and hypoxia, but not so clear the recovery in this type of sensors. You can find more information in our paper related with this research. And currently we are working also with other different implantable sensors. We are in a Pathfinder European project where we are injecting directly in the vessel implantable electronics readouts that are directly wireless to external from the externally to the vessel, and we are integrating aptamer beacon systems for detecting different proteins related with cardiac disease. And also we are doing a proof of concept of advanced in vitro models where we are mimicking the tissue of the brain in a chip, and we are integrated also monitoring systems inside this brain on a chip system. So thank you for your attention and sorry for the technical issues. And if you have any question, I will be open for answer it. Thank you.
Much, Dr. Krishna, for the insightful presentation. It was really very informative. So let's see if there's any question from the floor.